Hello. In this presentation, I will introduce the basics of gas emission source localization using autonomous science. This title might seem complicated, and that's because it is complicated. So let's rephrase that, shall we? We can also call it, Can a Robot Find Who Farted? And even though this title might seem crass, it is actually quite accurate, as we'll see later on. So let's start. Why is smelling so important? As David Bowie famously asked, is there life on Mars? And these two questions hang quite closely together. So there are a few indicators of extraterrestrial life, and one of these indicators is the presence of methane. And methane is actually an odorless and colorless gas, but we can still detect it, and we can use an analog of smelling with robots to detect methane. NASA's Curiosity rover actually smelled some methane on Mars. It was actually on 4 occasions in 2013 and 14, and it detected relatively intense concentrations of methane. So why does methane indicate life? We say this theory that methane is an strong indicator of life because here on Earth, most methane in the atmosphere actually comes from life, mainly from anaerobic microbes called methanogens, so methane generating microbes. And of course, we all know the well-known stereotype or joke about the cows on the pastures burping and farting methane gas into the atmosphere. And methane, of course, also contributes to the global warming and the greenhouse effect, so it is a very critical part of our atmosphere. So, what does life smell like? There are a few different origins of methane. So we have the first main group, which are non-biotic serpentine deposits. So these are the sources that are not directly linked to biological activity. What this means, non-biotic, of course, that it's non-biological, and serpentine is a type of rock formation that contains trapped methane gas in itself, in its structure. These deposits can then release the methane into the atmosphere through natural processes such as geothermal activity or geological disturbances like earthquakes. So these are not indicators of life. However, we also have subsurface methanogens. These, as the name implies, are located under the surface, and these have a biotic origin, so it relates to the biological process of methane generation. So these are microorganisms that produce methane as a byproduct of their metabolism, and these are quite common on Earth here. There's a lot of microbes in wetlands, rice paddies, and inside the digestive tracts of animals, like the cow, as pictured. And we also have methane clathrates. These are also known as methane hydrates, and these are another form of biotic origin methane. And these are ice-like structures, so they have a crystal-like structure, which is composed of methane molecules trapped inside this lattice of water molecules. And these clathrate or hydrates form in cold environments such as permafrost or in the ocean, deep inside the ocean. And depending on the environmental factors, change of pressure, again some geological activity, these also can dissociate and release the methane gas. And this is also one of the largest sources of methane here on Earth. So these are the three main types, and if we manage to detect methane, then there's a high chance that it's actually of biological origin. So I don't want to bog you down with all the chemistry details because that's outside the scope of this lecture, but I will give a quick introduction. So there's this long, complicated phrase called chemolytic autotrophic methanogenesis. So what does this mean? Let's break it down into pieces. So let's start off with the first word, chemolitho-autotrophic. Chemo is easy, of course, that means that it relates to chemistry somehow. Litho means rock or stone, so it has something to do with rocks. And then autotrophic, auto of course means self, and trophic means and so autotrophic together means that organisms can synthesize their own compounds from inorganic sources. So let's build it up. Litho refers to the fact that there are inorganic compounds as the donors, as the electron donors into these chemical reactions. So the autotrophic can convert by itself inorganic matter into organic matter, and chemo means that there are also inorganic chemicals as energy sources. And what is methanogenesis? That one's a little easier. Methano, of course, refers to methane, the gas, and genesis refers to creation, a rather biblical word, but it's quite simple in our case. Methanogens are microorganisms that carry out methanogenesis, so they convert various carbon compounds into methane as a byproduct of their metabolism. And in chemolitho-autotrophic methanogenesis, certain specialized groups of methanogens use inorganic compounds as both the energy source and the electron donor for the production of methane. So there are a few well-known chemicals that can take part in these reactions. These are, as I wrote on the slide, carbon monoxide, so CO, hydrogen, H2, water, H2O, of course we all know that, carbon dioxide, CO2, and we can see a few of the reaction equations here. So in the first equation, 4 carbon monoxide molecules combine with 2 water molecules, and these create methane, that is the CH4, or methane, and it also releases 3 carbon dioxide molecules as a byproduct. There's also another variant where 4 hydrogen molecules and a carbon dioxide molecule reacts, resulting once again in methane and 2 H2O or water molecules. And on Earth, these organisms are located in various environments, and quite extreme environments too, such as deep sea hydrothermal vents, under the surface or subsurface, geothermal areas, and these play a crucial role in our entire functioning and in our life and our entire ecosystem. So, what are the abiogenic methane origins? There are 3 major hypotheses for where methane could originate in an abiogenic manner, and we can exclude some of these theories. So volcanic emissions are one of the main sources of methane here on Earth, but the Mars Odyssey Themis mission mapped the entire Martian surface at a 100-meter resolution in the infrared region, and it detected no significant volcanic activity. And as I mentioned previously, these gas hydrates, these ice structures locking in methane, are also thought to not exist on Mars because they require a very specific set of environmental conditions, high pressure and low temperature. So these are quite unstable as molecules, and they would probably be instantaneously dissociating if they were to come into being. And even if they do exist, they're underground, so we couldn't detect them easily. So our main hypothesis currently is the weathering of serpentinites. These are minerals such as fayalite, consisting of various iron and silicon molecules that contain methane inside, and during environmental weathering they release methane into the atmosphere. And one of the minerals is also olivine, which is widely believed to be widespread on Mars. So that was it for all the chemical, biological, geographical background. Let's go on to the robotics side. So what we just did is called expert system knowledge. Basically, when we send an autonomous robot to outer space, we aren't able to micromanage its every step. We need to trust it. But for that, we need to give it the necessary background knowledge. So just like I did in preparation with these beginning slides, we need to pack all this data so the robot itself can make informed decisions. And for this, there exist various models that have already been used, such as MESIS, which is a planning and scheduling system. But there are very many novel types because machine learning is becoming ever more popular and ever more widespread. So there are many competing ways on how to get a robot to autonomously execute its required experiments and analyses. So autonomous science, as I explained before, it's a very complicated subject, and there are various methods, such as a very popular one is unsupervised clustering, which is a machine learning algorithm family that, for example, could automatically categorize detected rock samples or detected chemical samples. And for all this, background knowledge is vital. And in the past, there have been entire missions with thousands of if-else rules, but of course this is not too elegant, so nowadays we're using more advanced artificial intelligence solutions. So how do robots smell actually? And this is now not a joke, so robots don't smell bad, but they do smell difficultly. Our nose is a very complicated organ, and a smell is actually a very complicated thing to describe. A scent is basically a small sample of the source material itself, because it consists of diffuse chemical particles just floating around in a gas. And these particles vary highly, both spatially, everywhere, and temporarily. Sometimes a whiff of a freshly baked bread is strong, and then it disappears. So when we create a robotic analog to our nose, we have to consider these many factors. For example, do we take an instantaneous measurement? Do we take a time averaged integrative sampling measurement? And we also need to collect all available directional information, various vectors of the source, or the carrier gas itself, or the source movements. So we have to use a lot of sensor fusion and combination of sensors. So here's a practical example. Zoe, the rover, was sent out to the Atacama Desert in Chile to detect chlorophyll. The Atacama Desert is one of the driest areas on Earth. It is a totally barren, inhospitable land, at least at the first glance. There are a few life forms existing, but definitely not on the surface. There are plant matters underneath the surface. So, according to our theories, this could be very similar to what we would find on Mars. So, the researchers sent Zoe to this place. As you can see, it definitely looks unlike most places on Earth and definitely looks extraterrestrial. And here's Zoe with her little measuring probe. That is a plow which dug beneath the surface of the Earth, uncovering and potentially extracting the plant matter. And on board, Zoe had various highly specialized equipment, such as a fluorescent dye sprayer, which could mark, dye and indicate the search chemicals. It had a lot of cameras on board, infrared spectrometers to perfectly analyze what it could find. And of course, Zoe was very smart. She had a Bayesian-based classifier to detect the biosignatures, and it was successful 75% of the time. It also used a Bayesian methodology to accurately map, trace these sources, whilst also localizing herself on this map. So this was complicated with SLAM. And she used wet chemistry to stain this with this fluorescent dye. So this would probably be quite hard to deploy to Mars. And of course, we concentrated on methane and not chlorophyll on Mars. And Zoe was also well equipped for anomaly detection. Using her background knowledge, her autonomous science data, she could identify anomalous samples which might prove interesting for later research. So putting it all together, we have to exactly find out who farted, so what is the actual source of the scent and where, so we have to localize it. And for localization, we need to model how a scent plume moves around, and for this we come to the very complicated topic of fluid dynamics. If the fluid, the carrier material, or here on Earth the atmosphere, has a low Reynolds number, that means it's viscous, it doesn't have a lot of turbulence, it has a smooth laminar flow. This allows for easy gradient descent based approaches, but this is more present in microscopic environments, because if we move to a macroscopic environment, of course air movement is a much more complicated topic. So we have a lot of turbulence, and it spreads these concentrated splumes into random filaments and patches, and we need to find the source for a random filament, which is a very complicated manner, but we're in luck. Engineers usually like to find a very elegant, lazy solution, so they looked back to biological examples. For example, the E. coli bacteria is a very simple organism, but as all organisms, it needs to eat. So the E. coli bacteria find its food sources using a random walk algorithm. If it's flagella, these little whips that it uses for locomotion, rotate counterclockwise, then it moves forward in a straight manner. However, if they rotate clockwise, it tumbles around randomly. So basically what it does is just tumble around until it detects a sense source, and then it moves straightly. And if it loses it, it again tumbles around. So as you can see, this is not a very efficient algorithm, and it's quite prone to detecting local minima, but Equali survived, so it's working. There are other examples. For example, the dung beetle. That moves around nearly always on a 2D plane in a zigzag motion. And this is a more robust algorithm, and it's less susceptible to concentration changes. There's also the silkworm moth, which flies around in 3 dimensions, which also uses a more zigzag algorithm, but in a more advanced manner. And we can model these movements using various Bayesian control algorithms. So we can use this prior existing methods to create source likelihood maps, or SLIM, and use Bayesian probabilities to continuously update the likelihood of the various sources. And of course we can combine this with SLAM and Kalman filters to further complicate it, or make it more robust, hopefully. So to summarize everything I just said, first off, methane is a high indicator of life. We can't say for sure that it's of biological origin, but it has a high chances, if we find methane, that there could be life on Mars, or there was life on Mars. And robotic smelling is more challenging than we thought. That's also a lesson we learned. We need to develop the sensors simultaneously together with the robust control algorithms to localize these scents. So the sensors need to work closely together with the control algorithms, and because of the large distances and the large time delays, and we need to ensure that this robot is capable of executing the experiments autonomously. So here are my sources. If you're interested, you can use these to look up the topic in more details.